Good afternoon, ladies and gentlemen, and welcome to the RENEURON Group PLC Interim Results Investor Presentation. Throughout this presentation, investors will be in listen-only mode. Questions are encouraged and can be submitted at any time via the Q&A tab situated in the right corner of your screen. Simply type in your question and press send. The company may not be in a position to answer every question it receives during the meeting itself. However, the company will review all questions submitted today and publish responses where it is appropriate to do so. These will be available via Investor Meet Company dashboard and will notify you by email when it's ready for your review. I'd also like to remind you that today's presentation is being recorded. Before we begin, I'd like to submit the following poll. I'd now like to hand you over to Olav Halibo, CEO, Catherine Isted, CFO, and Stefano Plicino, CSO. Good afternoon to you.
Thank you very much. Welcome everyone. I look forward to spending the next hour or so with you. Let's jump into the presentation. There will be some opportunity for Q&A later on. So welcome to the interim results for the 6 months from April through September for RENEURON. Let me introduce to you Catherine Isted, our new Chief Financial Officer. She joined us in the middle of October from Oxford Biomedica, where she was in the finance leadership team heading up corporate development and investor relations. And I'll also introduce Professor Stefano Plicino, our CSO. He joined us in May, so 6 months in already. This is the first time that he's doing one of these calls, so I'll introduce him today. He is a professor of regenerative neuroimmunology at the University of Cambridge and a world-leading expert on exosomes, which comes in very handy for us. So with that, a quick disclaimer on since we are a public company, so I'll leave it there for your reading. And then we'll jump into a quick overview of RENEURON. So we have deep experience in the areas of stem cells and exosomes. Our most advanced program is in retinitis pigmentosa, where we expect data from the extension study in late Q1 next year. We are expanding our exosomes platform. We have now 7 ongoing collaborations with big pharma and significant biotechs, as well as academic institutions. Our financial foundations are solid. We have £17.4 million in cash, so that secures us at least a 12-month runway from today. Our strategy is to leverage our expertise for both products and technology platforms. HRPC is exciting, as we're talking here about an off-the-shelf medicine with the potential to reach a broad patient population. I will get much more into this later. There's a lot of value in our platform technologies, especially true for exosomes, where we have significantly grown the number of ongoing collaborations. And I would like to highlight that we are the only company in the exosome space that match the exosomes with the target tissue type. And we will come into much more details on that as well later on. But before we get there, let's have a look at the highlights for the period. For HRPC, we have continued to progress the phase 2A extension study. After a temporary suspension, enrollment was restarted in all geographies in October. We expect enrollment to be completed by the end of the year with data in late Q1, as I mentioned. The exosomes platform is expanding. We have 7 collaborations, as I mentioned. We have achieved proof of concept showing that our exosomes can deliver a therapeutic protein to the brain. This opens up a number of important neurological diseases as potential therapeutic targets, and Stefano will share that data with us a little bit later. We expect to deliver further proof of concept data from our existing collaborations as well in the year ahead. For the IPSC platform, we have initiated a collaboration with Dr. Claire Roddy at UCL on generating CAR Ts and NK cells from our iPSCs. In another UCL collaboration with James Phillips, our iPSCs have already been differentiated into Schwann cells, and they have a potential application in repairing peripheral nerve damage. We've made important changes to our board and management teams as well, and I'm very pleased with. Ian Ross has joined us as chairman, Barbara Stalin as senior independent director and head of the audit committee, and I already introduced our new CFO to you. But I'd like to add to what I said earlier, that I'm glad to have someone of Catherine's caliber on the senior management team. She possesses a great combination of scientific and financial knowledge. There are not many accountants who have worked as bench scientists, so we're very happy to have her on the team. So over to Catherine for the financials.
Thank you very much, Olaf. That's very kind. First, I just want to say, what an exciting time it is to be joining a company such as RENEURON. In terms of the retinitis pigmentosa program moving ahead, but I think particularly also on the exosomes program, which we're going to be coming into a lot of more detail later on. It really is, right time, right place for this technology, as we see a growing market in exosomes, and I really see and believe that RENEURON is leading the way. So on to the financial highlights for the first half of 2021. Revenue for the period was 58,000, relating to royalty income. The loss for the period was 5.2 million, so that was lower than last year of 7.1 million. This is driven by lower costs. On the cost front, R&D was 4.3 million. A decline of 1.6 compared to the prior period. This was based on the decision previously to not progress the stroke disability program internally, and instead focus resources on the company's RP program and our exosomes and IPSC platforms. G&A also declined in the period slightly. We had an increased net cash used in operations of 4.6 million, but that is more reflecting the lower figure in the prior year, where we actually received the 2019 tax credit in this particular period. So hence the cash, we have less cash used in that particular period. And then finally, as Olaf mentioned earlier, we have cash in the bank of 17.4 million, and that provides us a 12-month runway. And I should say that's a 12-month runway from today, as opposed to the date of the accounts. I'll now hand over to Stefano to go through exosomes in more detail.
Thanks very much, Catherine. So I think I have to get started from the very basic of exosomes in order to give the audience a clear idea of what exosomes are and how RENEURON aims at diversifying from potential competitors. Next slide. Okay. So exosomes is something that Mother Nature has provided every cell, every eukaryotic cell with. They are nanovesicles which are released continuously into the extracellular space, and they intrinsically traffic a number of potential conveyors of very, very complex information between cells. And what is being done in the technology space by different companies is to make good use of what Mother Nature created to develop a fully natural, fully biologic, new generation mean of delivering potentially therapeutic molecules. There is very established evidence out there that with exosomes we are able to deliver nucleic acids, and we're also able to deliver biologically active proteins and peptides. So this is not new. But what is new at RENEURON? RENEURON possesses a number of clinically relevant and biologically relevant cell lines from the brain, from the retina, and from even pluripotent iPSC lines, which provides the company with a unique opportunity of developing a world-leading repertoire of proprietary assets with potential application in a new wave of precision medicine approaches. We make the strong claim of being able to produce tissue-for-tissue exosomes. Plus, we have a very extensive internal expertise to help with the end-to-end processing and of exosomes, from the initial production of cells to the isolation to the characterization and manufacture. Next slide. So the technology that we are discussing today is a new wave of drug delivery technology, which as its own foundation in the possibility of delivering wide range of payloads using exosomes. And it has been extensively claimed by other companies, which is nucleic acids, soluble protein, protein associated to the membranes, and including also entire gene therapy vectors. What RENEURON is different about is the potential to produce complete tissue matching between the parental producer cell line of the exosomes and the final tissue to target by means of this new type of drug delivery technology. Furthermore, RENEURON has the capacity to use external loading to further assist in targeting the correct tissue. We are able to functionalize our own exosomes with 2+ payloads, which can be either incorporated into the inner compartment of the exosome, so even carried externally. And also, we rely on the extensive evidence of a very, very good safety profile, as well as little immunogenicity of exosome compared to parental cell line, as well as their capability of being redosable and reinjectable. So these are a number of key features which all point towards an extraordinary versatility of the exosome technology platform, which, if combined with RENEURON potential of making fully tissue matched, really go back to the initial statement made by the CEO and CFO, which is the right technology at the right place at the right time. Next slide. Right time, yes, the time is now. There is growing interest in the market, which is still growing towards exosomes. There is growing interest and focus in the scientific community towards developing more and more applications of exosome-based therapeutics. And as a matter of fact, as a byproduct of this growing market and growing interest, even RENEURON has significantly increased the number of academic and industry collaboration that we have developed in the last 3 years. You see that we moved from only 2 collaborations in 2019 up to 7 collaborations right now. You can see there that are listed 3 alliances between competitors: Evox Takeda, Kodiak Jazz, and Evox Lilly. Only one, Evox Lilly, is targeting neurological disorders and neurological diseases like RENEURON. All the others are either targeting rare diseases or cancer. So there is lots of interest. The interest is increasing, and the commercial value and potential of exosomes and exosome-based deals is increasing as well. Next slide. So what is the advantage of our approach? On the left of this slide, you see what the competitors used to propose. They used to start from a single, fully characterized, stable cell line. Usually they are cancer cell lines. And to apply cellular or exosome engineering approaches to the exosome produced out of that individual cell line, proposing targets without any claims towards tissue matching. So they work with single cell lines. They identify multiple clinical indications. They don't claim. They don't care about tissue matching. What RENEURON instead is proposing is a novel tissue for tissue approach, or precision medicine approach, by which using the different cell lines that we have in our portfolio, starting from neural, moving to retinal, and then ending up with pluripotent stem cell lines, to develop tissue-precise exosome therapeutic with full tissue matching with a final tissue to target for our indications. Next slide. How do we do this? We do this by combining very extensive knowledge of the cell lines that we have developed in the course of the years. We have several brain stem cell lines from the cortex, from the striatum, and from the hippocampus. They are producer lines which have been extensively characterized. They are also conditionally immortalized, and this conditional immortalization feature provides these cells with the unique feature of being much more stably expandable than non-conditionally immortalized competing cell lines. They're also GMP compliant, and the GMP compliance can also be applied to human retinal stem cells and human retinal stem cell exosomes, which all the promise of being potentially superior in the contribution to the target cells as compared to parental cells. We also have a very early stage technology platform, which is based on our proprietary CTX cell lines, which is a neurostem cell lines that researchers at RENEURON have managed to successfully reprogram into a stably expandable IPSC line, which is pluripotent in nature, and which has therefore the potential to produce a multitude of tissue-specific, stably expandable intermediate stage progenitor cells for production, for downstream production of exosomes, which opens the opportunity for a completely new era of fully personalized tissue-specific exosome therapeutics. So it is a very exciting repertoire of assets with huge potential for applications in a number of diverse therapeutic areas. Next slide. The way we modify our exosomes is very, very exciting. We apply the principles of passive or active loading of exosomes, as compared to what other companies are proposing as native, unmodified exosomes, by therefore only using the intrinsic potential of exosome continuously produced by cells. So the passive loading is very simple. Basically, we collect exosomes out of tissue culture media from producer cell lines, and we apply sophisticated means of exosome engineering to the exosomes in order to increase their capacity to deliver specific payloads. Usually, the passive loading is applied to payloads which have to be carried on the membrane of the exosomes, and which applies to specific clinical indications. If instead the case is for making exosomes smart enough to be able to deliver intra-exosomal payloads, then we apply what we call active loading or cellular engineering protocols, by which we modify the producer cell line in a way that will produce appropriately modified exosomes with specific payloads in their inner compartment. Combination of passive and active loading provides RENEURON with a hugely expanded capability of producing exosomes for several clinical indications. Next slide. And this is what the CEO briefly mentioned at the beginning of our webinar today. Our portfolio of collaborations has increased significantly in the last few years. We now count a total of 7 academic or industrial collaboration, among which you can identify here 2 global pharma, 1 large, 1 medium, 1 small biotech, and 2 university collaborations, all of which are fully diversified in terms of payload to functionalize onto exosomes, all of which have shown different pace of progression from discovery to proof of concept studies, and all of which hold significant potential for transition into in vivo late stage projects. We also have very exciting internal or corporate programs, among which using exosome to deliver microRNAs, which is in vitro at the moment, using exosome to deliver gene editing technologies, including CRISPR and Cas9 enzymes, and also using exosome to deliver therapeutic growth factors. And the reason why I have listed as last this ExoGF program is because I'm going to show you in a moment a very exciting set of proof of concept data that have increased substantially the enthusiasm at RENEURON towards the possibility of delivering therapeutically sound protein molecules in vivo into the brain using exosomes. Next slide. So I've studied exosomes under my academic hat for more than 15 years, and I have to be honest, I've never seen such an exciting proof of concept set of data in vivo. The experiment that we ran here was aiming at delivering a specific growth factor, which is called BDNF. BDNF stands for brain-derived neurotrophic factor. It's a neurotrophic factor which has been extensively studied for its capacity to modulate neuronal and astroglial functions in the brain, specifically into the brain. And when dealing with drug delivery technologies, even old-fashioned or new generation drug delivery technologies, the brain has always been a nightmare as a site of preferential targeting. So in this specific context, we used the thecal injection via lumbar puncture route to access the cerebrospinal fluid. So it is a minimally invasive approach whereby exosomes are injected directly into a biological fluid which naturally spread over the entire brain and spinal cord. And we compare in vivo the capacity of signaling towards its cognate receptor of unmodified exosomes, the native protein as it is coming from commercial sources of protein-loaded exosomes, which you can see in light purple in the bar graph. And what we measured 2 hours after the injection was the expression of an early gene, which is usually expressed only after that protein is able to bind to its cognate ligand onto target cells. And you can see how much is the difference and how much efficient, between 3 to 4 times more efficient were those protein-loaded exosomes in achieving the type of signaling which is reminiscent of a therapeutic outcome. The therapeutic outcome is not there yet. We have filed a patent application earlier in October this year. And the next step, which is what we are excited about, is to perform a number of studies, all functional, in vitro and in vivo, to demonstrate what is relevance of delivering PDNF to a much higher extent of signaling into the brain in laboratory animals with specific disease models. BDNF is relevant because it might be applied to diseases like Parkinson's and Huntington, for which there is no cure yet. I think I'm done with the exome presentation, and I'm happy to pass it to the CEO as well. Again.
Thank you, Stefano. Yeah, so let's move over to the retinitis pigmentosa program. So RP is an inherited eye disease, close to 100,000 patients in the US. So it's orphan, but it's a large orphan, if that makes sense. There are more than 100 different genetic mutations that lead to RP. So that means it's not a great disease to use gene therapy. You're not going to solve all of those different gene mutations. With gene therapy, it's a very fragmented market. Having said that, there is a gene therapy available, which is Luxturna. It is available for patients with RPE65 gene deficiency, but for the other 98 or 99% of patients, there's nothing available for the moment. One benefit of using retinal progenitor cells that we do is that they have the potential to benefit all RP patients, no matter the cause of their disease. So it's a gene independent. We've shown in animal models that our HRPCs differentiate into photoreceptors and integrate into the optic nerve. Integration is likely to provide a durable trophic support also. You need to realize that the photoreceptors, you're born with all the photoreceptors you'll ever have. So if you lose them, the body will not create new ones. So, being able to recreate or create photoreceptors would be really the holy grail here. The program was licensed in from Skeppen's Eye Research Institute at Harvard, and we still work closely with them as well as with UCL and Moorfields here in the UK. The invention of the IP-protected low-oxygen manufacturing culture has enabled us to produce the cells in large quantities. So this breakthrough was really the foundation and enabled us to grow the cells enough to be able to make them into a potential medicine. The development of cryopreserved final product formulation also has been very important and enables us to ship and treat patients globally. We can deliver the vial to the patient rather than patients having to go to where the manufacturing is happening. The commercial potential for this is without doubt very large, particularly for this, since this is gene independent. The gene therapy Luxturna is launched, as I mentioned, and is priced at $850,000 per patient in the US. 615,000 pounds is the NICE approved price in the UK, so it's similar pricing globally. So in a 12-patient phase 1 study, we established safety and advanced from fresh to cryopreserved cells. And what you see here is our phase 2A study. We treated a further 10 patients. They were all treated with a dose of 1 million cells. and the chart at the top right here shows the results that we collected so far. So 9 patients had successful surgeries, enabling them to benefit from the cells. Some patients have not been able to come in for pre-specified visits due to COVID, so that explains why the N drops from 9 to 7 between 6 and 12 months. It's patients not coming into visits. 6 months is the primary endpoint in the protocol, but we do expect FDA to be asking for 12 months data in later studies, so 12 months is important as well. The key measurement in ophthalmology studies is the number of letters read on an eye chart. So the chart has 5 letters per line. If you could read every single letter, you read 100 letters. And if you read less than 35 letters, you are legally blind. And if you read 85 letters, you have 20/20 vision. So there's 50 letters from being legally blind to having perfect vision, just to give you a kind of feeling of the scale. So a few letters are very important. Having said that, any improvement or even a stabilization of vision is valuable, because this disease, with this disease, you're on a path to a total loss of vision. It is an unavoidable event. So even having any impact will be extremely important to patients. So having said all of this, if you look at the chart, the top line there is the change in vision compared to baseline in the treated eye, and the bottom line is the untreated eye. So we treat 1 eye for the patient and not 2. We have observed a rapid effect in most patients, resulting in a sustained effect overall the time points, including the 12-month one. We're encouraged by the rapid and sustained effect of around 8 to 10 letters, as you can see here. With so few patients, it makes sense to look at the individual patients, and that's what we're doing here at the bottom right. We take in the 12-month data point and the 7 patients that we have data for, and you notice that all the patients had some improvement from baseline, some in the 6 to 8 letter range, and 3 patients actually above 10 letters, which is really fantastic. That's an improvement by 2 lines or more on the eye chart. Now, as we have promising efficacy result, but no dose-limiting toxicity, we expanded the study to treat an additional 9 patients with a higher dose, and we call that the extension study. So this is double dose of 2 million cells. So we would like to see if this could give additional benefits, particularly to those patients who were on the left-hand side, the lower end on the previous page. We also added some additional efficacy measures in consultation with our opinion leaders, and we have added 3 additional clinical sites. Professor McLaren's sites in Oxford is one of them. We are currently recruiting the remaining patients in the extension study, and we look to complete recruitment by the end of the year. That means we'll have some early efficacy data in late Q1 next year. So analyzing this data will inform us then on kind of what's the right dosing, 1 million, 2 million, how does the commercial profile emerge from this, and then what kind of study will we embark on next. And it will also inform us on the best route in terms of partnering and licensing this drug going forward. So a quick roundup on IPSC and CTX as well. So we are also very excited about the opportunity we have with the iPSC platform. We have reprogrammed our neural stem cells into a pluripotent state, which means that they're able to differentiate into any other form of cell. Now, while doing this, our iPSCs have retained the immortalization from CTX, which results in highly stable cell lines. So immortalization means that you don't need to rederive, and you can make very large quantities, which obviously has a huge advantage. There are 2 collaborations with UCL ongoing, one investigating the potential use of CTX iPSC cell lines to generate CAR Ts and CAR NK cells. And in the other one, we have already succeeded actually in differentiating our iPSCs into Schwann cells, and that means we have a potential use there in peripheral nerve repair. We will continue to expand the work we do in this very exciting area of science. For CTX, it's being pursued by our partner, Fosun Pharma in China. Stroke is a very large issue in China, mainly due to smoking, high rates of smoking. So it's a very large interest for Fosun, so they're pushing this ahead. There are other potential indications also for CTX, well supported by preclinical data that Fosun is interested in looking at, but we have not started any formal work on that. We will continue to look for a partner in other geographies as well for CTX. So I'm going to do a quick summary and then we'll go into Q&A. So for exosomes, we are in the right place at the right time with the right technology. We may have been too early with our stem cells, but it has enabled us to now be ready and hit the sweet spot for exosomes. We are accelerating our investments and creating that know-how and valuable IP in that area. HRPC has a near-term potential to build near-term value, and we're looking forward to the data and the next steps there. And IPSCs as well as CTX are upsides to build long-term value. So we're very excited about the future, and I look forward to your questions.
Olav, Catherine, Stefano, thank you very much for your presentation. Ladies and gentlemen, please do continue to submit your questions using the Q&A tab situated on the right-hand corner of your screen. But just while the company take a few moments to review those investor questions that have already been submitted, I'd like to remind you that a recording of this presentation, along with a copy of the slides and the published Q&A, can be accessed via your investor dashboard on the Investor Meet Company platform. I'd also like to remind you that your feedback is important to the company, and immediately after the presentation has ended, you'll be redirected for the opportunity to provide your feedback in order that the company can better understand your views and expectations. We received a number of questions from investors, and if I may, I'll read the first one. Will shareholders get any news on actual data obtained in these collaborations on exosomes, or will we wait until any licensing deal is signed?
Yeah, so if I understood the question correctly, so this is about exosome collaborations. We obviously work with partners on them, so there's always a question of having a discussion with a partner what is released when. Having said that, when there is material news for Reneuron, we will share that with the market. We might have to disguise it depending on the discussions with the partner. So it might have to be product X rather than exactly what it is. But we understand that there is an obligation to report that to markets. So that's on the data. We do also hope/expect that proof of concept data will then eventually lead to a deal if it is with one of the pharma partners. And then obviously when the deal is signed, that will be announced immediately on signature.
Thank you very much. The next question reads as follows. Within the next few years, do you anticipate Reneuron listing on the NASDAQ?
So fortunately or unfortunately, it seems to be the goal of every successful European biotech to have a US NASDAQ listing. So, we're not different. We have the same ambition. So, but I think our immediate focus is to have success in the UK.
Thank you. Are you in discussions to license IPSC platform to larger pharma companies?
No, it's probably a little bit too early. I think we're still developing that technology, and I think this kind of academic collaborations is the right way forward for the moment. And I see Stefano nodding. So
Yeah.
So then that's what's going to happen.
Thank you. The next question reads as follows. Do you think Reneuron will use the 1 million or 2 million cell dose for their pivotal phase 3 approval study?
I'll let you know in March.
Could you clarify the ownership of your cell production suite and what its capacity is as you start to produce cells for partners, i.e. is further upscale investment required?
So depending on the platforms here, so for the clinical grade materials, so for HRPC as well as CTX, they are produced with CDMOs externally. Exosomes, we have the capacity to produce those now internally. when you come to kind of further down the road, and you want clinical grade material, we have to make a decision if you want to invest in our own GMP manufacturing capacity or work with partners on that. So it's a political answer, but we kind of have two routes on that.
Thank you for that. Does the exosome platform benefit from lower regulatory burden as it is derived from CTX cells that are already approved for trials? If so, how could this be seen in trials design?
So I think if I'm reading the question correctly, so our exosomes are derived from CTX. So some of them the basic or the original exosome we're starting with exosomes from other tissues as well, and from iPSCs that Stefan was explaining. But the CTX derived exosomes obviously come from a clinical grade starting point. So that's helpful. It's also very highly valued by partners because that they know that is a stable, highly analyzed line that we've reproduced many times. I'm not sure it's going to change anything later on in terms of clinical trials. It might just mean that it's a little bit easier to get there.
Thank you. The next question asks if you would consider running any phase 3 HRPC trial in-house if the funds became available through a commercial exosome licensing deal, or would such funds only be used to further develop the exosome and IPSC platforms?
I obviously can't kind of answer categorically about how we intend to use funds going forward, but we always look at there's always a balance between partnering a program where you share the future revenues and profits for the partner, but you lower your risk versus keeping everything for yourself with a higher risk and then higher potential future return. It's always a conversation with shareholders. And it also depends on what deals are on the table. So again, slightly political answer, but it's because it has to be that way. We're open to both of those approaches. We'd love to create an important long-term viable biotech company based in the UK, but that doesn't mean we're not going to do lots of deals.
Thank you for that. The next question reads as follows. Because my wife has early stage Parkinson's disease, I am personally very interested in the huge amount of research work worldwide on the use of stem cells for treatment, many with apparent success. Is RENEURON involved with any partners in this work, and in particular the possibility of finding a delivery system for the dopamine drug to be carried across the blood-brain barrier? This would seem a great application for the company's exosome product, and as this is a potentially hugely profitable market, I'd like to know of any current or future plans to become involved by the company.
Yeah, I agree with the person that Parkinson's disease is a very important disease to treat, and it's a difficult one to treat. I'm not going to speculate what indications we'll end up choosing. It will be a balance between where the biggest unmet medical need is and where we think we can make the biggest difference versus what else is already available or in terms of being developed. So, Parkinson's disease, clearly, an important CNS disease, as is Alzheimer's, Huntington, and a few others. So, that's a process that we go through internally. And when a decision is made and the program is started, we'll announce that.
The next question asks, when will trials start in China with Fosun regarding strokes and RP?
I will let you know when we have news to share with you. China is such a big market and such a big opportunity, and all I can say is that I'm very happy that Fosun is very much fully behind it and progressing it and pushing it forward. But we have not started a clinical trial in China yet. And I don't want to speculate on when because it's slightly out of my hands. So I can influence, but I can't direct.
Thank you. Do you plan to fund HRPC phase 3 trial, or will you bring a partner in beforehand?
It's sort of the same answer as before. We will look at both.
Thank you. With regards to the ongoing pharma exosomes collaborations, we will get notification of any proof of concept achieved along the way. Or will we only get an update when the collaboration either ends by way of a licensing deal being signed or both parties terminating the collaboration?
Yeah, I think that was asked in a different way earlier. We will announce any material data, even if we have to disguise them. And we will obviously announce deals as they are clearly material.
Thank you for that. The next question reads as follows. In the expanded HRPC phase 2A study, how many of the 9 additional patients have received treatment to date, and when do you expect the last patient to be treated by?
Yeah, so we don't comment on patient-to-patient kind of data, but our expectation is that we'll have the 9 patients treated by the end of this year.
Thank you very much. When do you hope clinical trials will start for our pancreatic islet B cells in diabetes?
Yeah, we do have a program ongoing there, but yeah, I think we're pretty far away from that still. It is something that we're working on to create those cells, and then after that, they will need to go into animal studies. So it's a long way, honestly, away from clinical trial. There is, I think Vertex has started with some clinical program with islet cells in diabetes. So it's great to see that stem cells are starting to make inroads into large clinical trials.
Thank you. Well, the next one isn't a question, but the attendee just wants to take the opportunity to congratulate the company on developing 2 platforms that could potentially lead to a major breakthrough in treatments available for those in medical need. If you had any comments on that.
Well, we say thank you very much for the comment.
Thank you for that. With regards to the progress being made in the ongoing HRPC trials, are there any third parties that we provide updates to that would potentially be interested in a future licensing deal?
We are under CDA with several companies for that program. So yeah, I would say there are several companies that are interested in the space and keep an eye on us for sure.
Okay, thank you. And just turning to the final question, how many active pharma collaborations are still ongoing regarding the exosomes?
So all the 7 that we presented are ongoing. And if you're asking for global pharma, there are 2 global pharmas, and there's still 2 global pharmas.
Thank you. I think you addressed all those questions that you can from investors. And of course, the company will review all questions submitted by investors today and will publish those answered on the Investor Meet Company platform. Olaf, just before concluding, I just asked if you could provide a closing snapshot for investors.
Yeah, be happy to do that. First of all, thanks to everyone for your interest and participation and for all your great questions. So with exosomes, we're on the right place at the right time with the right technology. And for that reason, we are accelerating our investments and creating know-how and IP in this area. With the HRPC program, we have near-term value potential, and we're looking forward to the data and the next steps there. And for IPSCs as well as CTX, they are upsides to build long-term value. So we're very much excited about the future. Thank you very much.
Olav, Catherine, and Stefano, thanks again for updating investors today. Could I please ask investors not to close the session, as you'll now be automatically redirected for the opportunity to provide your feedback in order that the management team can better understand your views and expectations. This will only take a few moments to complete, however, is greatly valued by the company. On behalf of the management team of RENEURON Group PLC, we'd like to thank you for attending today's presentation. That now concludes today's session, and good afternoon to you all.
Thank you.
Thank you.